Everybody, here we are doing a quick little video for this lesson on genetics. So here we go. I just want to quickly show you guys where we're at here. Let's see. This is what I need. Pull it up. So I'm in the process of making all this stuff. So what you guys will see, here's the entry screen. If you right now, if you're here, you're in unit number 2, probability and genetics. You've worked through this right here. You've worked through this, and now you're right here. So we're just kind of exploring these concepts of basic genetics. This will be a review from biology, but we'll do it nonetheless really quickly to help you get through that. What you guys will need to do when you click on this, it'll pull up. I will have the video here that I'm just making right now. It'll be posted right there, plus a few other resources that you'll need to read through. And what you guys are going to do is you'll work on this genetics guided notes. I want you guys to take notes on this. You'll actually submit this to me to show me that you understand this stuff right here, okay? So just basic terms that you'll need to know for the test. And these 2 laws I'll use a lot, okay? I'll also go over the product rule and sum rule in an article that I'll have you guys read. And then number 5 is, and 6 and 7 are practice problems for this, okay? So pretty simple, pretty speedy on that. And this is what we'll do for day 1. And then day 2, I will have a lab and things that you guys will start working on. All right, so to get started, let me pull this up. Let me get just situated here, and let's swap that. Okay. All right, you guys, so if you guys remember, this was my video, or my thing that I started the unit with was this. So here's these 2 girls. They are actually twins, if you didn't watch the video, and they came from the same mom, same dad. And so to me, I think, wow, well, how is this happening? What's going on here? What's causing this to occur? And to get started, I want to play a quick little video basically looking at what are traits. Here are different traits that we're seeing, extremely different traits that are coming from the same parents that we're seeing expressed in this family. Okay? All right, so here we go. Let me pull up the video.
We are all unique. Each of us has a combination of characteristics or traits that is different from anyone else's. Some traits are easy to see, like eye color, height, and freckles. Others are less visible, like asthma, musical talent, and disease risk. Some traits are inherited, and some are shaped by the environment. Eye color and nose shape are inherited traits. They are controlled by genes, which we inherit from our parents. The language we speak and the music we like are shaped by the environment, which includes our culture, our experiences, and our physical surroundings. Most traits are influenced by both genes and the environment. For example, tall parents tend to have tall children, but height is also influenced by nutrition. And while diabetes tends to run in families, some types can be controlled by diet and exercise. Even when someone is born with a body type suited for dancing or playing basketball, it takes training and practice to learn these skills. Together, our genes and our experiences make us who we are.
Okay, so thank you, University of Utah, for making that lovely video for us. So you guys, from this, you can actually work on your note catcher here. Traits. What are traits? Okay. And so that video kind of goes over that. What is a trait? We can see that there's things that will influence traits differently. We know that it's genetically inherited from mom and dad, that comes back from their parents and their parents before them. So we can see that there's trends and patterns that we can see there. But also, we can see that environment also plays a huge factor in how traits are inherited. All right, let's go back to our PowerPoint here. Why does it do that? There we go. Okay. So the next video that I want to play is just looking at genes. Okay, how are genes different?
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order. But small differences in the sequence of the bases in our genes make each person unique.
Okay, so again, thank you, University of Utah, for their lovely video. Okay, so you guys, from that, you can see just how unique we all are and what's actually causing those traits to be different. Okay, so it's the changes in our genome that's causing that to occur. So you guys, today, really fast from this video, recording this lecture, you'll be able to identify and describe these different terms. And yeah, so let's get started. So first, to start out, I like to go briefly into just Gregor Mendel, and what he did. Basically, he's the father of genetics. He's the one that discovered a lot of things, got us started to think about that traits are inherited and are passed down from parent to offspring. And how he did that is he was actually a monk and lived in isolation by himself a lot, but he liked to garden there. And in his garden, he grew pea plants. And what he would do is he would look at different traits. There was actually 7 different traits that he looked at, and I'll show you guys those right here. So he looked at peak pod shape, at color, at height, flower position, flower color, seed color, and shape. And what he was doing is he actually saw several things here, and I'll kind of describe these. But as he was testing these, he noticed that there was patterns that he was able to see when he was crossing these. And it actually shows you right here one example that he kind of did. So you'd take some plants. He had a tall plant and a short plant. He would cross-fertilize between the two. And he found out that the F1 generation, or the children, the offspring from the parent generation, the P generation, were all tall. And he was like, okay, well, that's interesting. So there must be a trait that overpowers the mass one, that's dominant to the other. And so then he was like, okay, well, what if I cross-fertilize the tall plants from the F1 generation, cross-fertilize? What happens then? And he saw something very interesting. He saw a pattern of a 3-to-1 ratio, 3 tall to 1 short. And he noticed, oh, look, that tall, or that short phenotype, that short characteristic, skipped a generation, but it's still there. And so he started to explore this a lot more. He came up with 2 laws, and here are these laws. The first one he calls the law of segregation. Now, the law of segregation is basically the idea that versions of a gene, so these alleles, the dominant or recessive alleles, are going to be inherited independently from each other. Okay? And an article that I'll send you will describe this, that will be posted, and so make sure you read that to really in-depth understand the law of segregation. But the law of segregation basically states that a parent will have 2 alleles for each gene, okay? And that parent can only pass on 1 allele to each of its offspring. So, for example, this parent right here, big R, little r, it can either pass a dominant allele or a recessive allele off to its offspring. It cannot pass on both. One comes from this parent, the other one would come from this parent. Okay? Same for all these. And so Punnett squares, they coined these Punnett squares. They show probability. They show all the different possibilities that can come from these two parents. Another law that he came up with was the law of independent assortment. Basically, the law of independent assortment says that that genes will inherit independently from each other. So, for example, right here you can see there's 4 letters. Right here there's only 2. We're looking at 2 different genes. We have this one right here that's determining one gene or one phenotype, one characteristic, and then we have this one right here that's posing another. And so, and you can see the same thing here. And so, one's for color, the other one, I just know this because I've taught it for several years. But, anyways, the Y's are showing color, so yellow or green, while the R's are showing shape. You can either be round or wrinkled. The one thing that I don't like about this is completely wrong. This should be a big R right here, big R. Let's fix that right now. Bad. So this should be a big R. Big R. Found an error in your video or in your picture there, my friend. Okay, so there we go. So it's big Y, big Y, big R. Big R was crossed with a big Y, little R, or little Y, little Y, little R, little R. We get heterozygous offspring. They then cross, fertilize, and we see a pattern. And so, anyways, the law of independent assortment shows that each parent can only pass on one allele for each gene. So you can see that right there. So 1Y, 1R. And that's that. We also see the different patterns that can occur here as you do that. And we'll go into how to do these a little later. All right. So right here, how did Mendel's experiments involving single factor crosses support the law of segregation? So let's first look at the law of segregation. Right here, we're looking at this. Basically, like I said, each parent can only pass on one allele to its offspring. And so to do a Punnett square on this, oops, I don't want the pointer, I want the pen. So if we did a Punnett square here, we could model what's going on. So we know that tall is a dominant trait, short is recessive. So for this one right here, to make them all tall, what would these genotypes have to be? Well, I'm going to use a T here. There's 2 ways to be tall. We could be big T, or little t, little, or big T, or big T, little t. To be short, there's only one way, little t. And according to this, we know since they all were tall, what one must it be? It's got to be this. And so we know that this parent right here was big T. This one we know for sure is little t. And so when we fill out the Punnett square, there we go. We see 100% all being tall, but they're heterozygous, right? And so then what they did is they took 2 of these guys and they cross-pollinated them to get this. So let's do this one. Let's do it in blue. So big T, big T, little t. All right, so there we see a 3-to-1 ratio. Did it match that? Yes, it did. Okay, so we have a 3-to-1 ratio. If we were really to break it down, we'd see a 1 to 2 to 1 ratio if we looked at the allele combinations or their genotype. Excuse me. All right, so how is this showing the law of segregation? Well, it's showing that each parent can only pass on one allele at each time. Okay, and so that's modeling that for us there, and the data supports it, and that's what's fascinating. Okay, so then the next one, how did Mendel's experiments involving 2-factor traits help support the law of independent assortment? So these ones are a lot more fun because they're bigger, and let's see if I can just make my hands like die trying to do this. I need like a drawing screen. You know what I mean? I mean, if they're going to make me teach online, they need to do that, right? That's what I'm thinking. Anyway, so what he was doing is he looked and said, okay, let's look at two traits. So let's look not only at P color, but let's look at its shape as well. And he was like, okay, and let's take one that's homozygous dominant or dominant for each gene, and let's cross it with one that's recessive for both genes. And he's like, okay, and let's see what outcome we get. And so what he did is he actually did this, and this was what he saw. Okay, and if we really take the ratio, it's following a 9 to 3 to 3 1 ratio when we're looking at the different phenotypes. So let's see how it kind of models this, right? And so to do this, just to get you guys some practice, there's one question on your note catcher where you have to practice what we call dihybrid crosses. And a dihybrid cross is when you have two genes here. There's two ways you can actually solve for these, and I'll show you both ways, okay? But I want you to be able to do two-factor crosses. So if we have our parents, we have big Y, big R, is crossed with one that is homozygous recessive for both. The first thing we have to do is we could draw our little Punnett square here. And this one, it's going to be a lot bigger. It's a 4x4. Man, skill. All right, and so then the first thing, this is where a lot of students mess up. They have to model the law of independent assortment. Remember, each parent can only pass on one allele for each gene, okay? But it's all random how these get passed on. So remember meiosis is going to bring on half the DNA. So maybe you got this one, maybe it gave that one. We don't know. It's all random, okay? So let's figure out what these gametes are going to be up here that will create the offspring. And I like to do the foiling method. So one combination that we could get is this. Another combination could be this. Another combination could be that. And the last combination could be that. Okay, so let's do the same thing at this one right here. So we have little y, little r. Little y, little r, little y, little r. And so I'm not going to fill this whole thing out because it would all be the same. But we have our big Y, big R, little r. Okay, and I like to keep the letters together. That way I can see if they're homozygous or heterozygous and that kind of thing. So we see that all of them followed that same pattern. And so what he did is he saw that in the F1 generation, they were all heterozygous. And then what he did is he took this one, self-fertilized it. And this is what he saw in the F2 generation, is he saw a ratio of 9 to 3 to 3 1 ratio, showing that these genes do pass independently. And the reason I say that is because if we were to do it as this for each one, we would see the same kind of ratio there. All right, so if we were to do a Punnett square for each gene separate, then we can really decide or get the same ratios here. And so, for example, we have big Y, little y, and I did not do that very well. But you'd see that they were all heterozygous. Right? So we have 100% that are heterozygous. If we did the same thing for this one, we would see the same exact thing for the F1 generation. And so you can see how this supports that because we would just combine these, right? Big Y, big R, little r. That's the outcome. Now, if it gets a little messier when you have like this going on, because we would have a 3 to 1 ratio for each one. But then you'd have to say, okay, well, this one could match up with that one could match up with that, this one could match up with that one could match up with that. And you'd get 16 different possibilities. And so that's basically what this is doing, just in a different way. Okay? Alrighty. So another way that we can visualize these is using what we call pedigrees. And I love pedigrees. I think they're really fun to solve. And we'll get some more practice with this later on. But if you look at this, first of all, couple ways to understand pedigrees. The squares are males, circles are females. We see horizontal lines like this. If they're directly attached, that shows a marriage, while this would show their kids that they've had. So they had 3 kids. 2 of them got married. This one did not. And they also had kids. Now you'll ask, okay, what does it mean to be shaded in? Well, shaded in means that they show whatever trait that we're trying to look at. Okay, so for example, we could say that this is Huntington's disease. Huntington's disease is a genetically inherited disease that gets passed on and causes your brain nerve cells in your brain to degenerate, to kind of disintegrate, go away, and it's very not fun for the person that gets that. It usually will come along and later on set. Okay, so when we look at this, what is the pattern that we're seeing here? Is it a dominantly inherited pattern of trait or recessively inherited trait? And so just by looking at the video, we can say it's probably dominant. Why? Because here's this one parent. The next generation, they had 3 kids and all 3 got that. And so we must say that must be a dominantly inherited trait. We also see it happening here and right here, where we see some things going on. We could actually solve some of this using what we know about genetics now. So let's pretend that this is a dominantly inherited trait, or that's what our hypothesis is. So let's do Huntington's disease. I'm going to have H for Huntington's disease and lowercase showing no disease. So no disease, Huntington's. Okay. And so when we're looking at this, since this father is not shaded in, there's only one thing that he can be, and we know that for all of them. What is she, though? Well, there's 2 things that she could be. She could either be this or this. Which one's more likely? Well, they had 3 kids, and all 3 had Huntington's disease, so it's probably this. But is there a likelihood it could be this? Yeah, there is. So we don't know for sure. So I'm going to keep it like that. Now these ones right here, what type of genotype are they going to have? And if we did a Punnett square for both of the options, because we don't know which one she is, we could then see what's going on. So let's do homozygous dominant right there. Let's do heterozygous right here. So if we did that, we see that 100%. Sorry, my kids are crying. Okay. All right. So you can see right here, all of them that have Huntington's disease, they all have the same genotype, right? So all these, they're all big H, little h. These ones right here, we have a 50% chance that they would be normal, and then 50% chance that they would have Huntington's. But we can see that they all still share the same genotype. So we know for sure that all of these are heterozygous. We know for certain of that because there's no way to be homozygous dominant in the offspring. Now this parent right here, do we know what she is? No. So she could be either or. And so you can see how we can kind of solve this. If we did the Punnett square with this, we know exactly what these are. These guys are heterozygous. And using pedigrees, then people can come in and say, okay, well, I had a grandpa that had Huntington's disease. What are the chances that I could have Huntington's disease? And so by solving these types of pedigrees, we can help them to understand that a little bit better and kind of make the deciding factors based on that. Okay? All right, a couple more things. I went over this in class, so I'm going to just briefly go over it, but jot down this equation if you need to still. And then I will post one of these pages on Schoology for you guys to access. You also have one in your notebooks, or should. Okay? But basically, when we're looking at pedigrees and Punnett squares and stuff, we like to really have scientific evidence that shows that our data is correct. And so to do that, we like to use statistics. And one statistical test that I've already kind of gone over with you guys in class before we left was chi-square analysis. So just a reminder, chi-square is equals to the sum of all your observed minus expected values that are squared. Divide that by the expected. Getting that will give you a value. And we always want to look right here at the 0.05 range using this column right there. And so then using your degrees of freedom. And when we do a lab here later, I will explain this to you. So I just wanted to briefly go over this. Just make sure you have this ready because there will be a lab that you're going to do here in the near future where you'll have to apply that. Alrighty. Okay. And so that's where I'm going to end it. Now I'm going to discard those. Let's make sure I've got everything that I need to cover for you guys here. Yeah, I think I did. So you guys, there will be a few things, especially this one right here, the product and sum rule. Make sure you know how to do those. There will be some articles that I'll post there with it that you guys will need to read to get the extra information if you didn't quite get it here. Do some practice problems. One cool thing is if you're doing this on the iPad, on an iPad or something, you could actually just draw right on here. If not, I think, let's see. If you guys that are doing it like on a computer or something like that, just go, if you want to just do it on a piece of paper and then just take a picture with your phone, you can just put the problem up here. Show me that you did it on here. Just like paste your picture in these blanks. Another option would be to just go to Paint. So, right, and you can do them and then just copy and paste that picture right in there so I can see that you guys can do these basic probability questions. All right. Okay, guys, if you have any questions, don't hesitate to contact me.